Hello guys and welcome back to Agriculture Academy. So today we are going to do organogenesis in plants. Again, it's a part of plant tissue culture series. So I'll be continuing with this plant tissue culture till it gets completed as per the GADB IIT JAM syllabus. Okay, so stay tuned with me. So cultivation of plants is using the organogenesis. So what is organogenesis? It's actually a process. So it is a process which involves redifferentiation of meristematic cells. Okay, so these can be either in the shoot. Okay, so these can be either from the shoots or from the roots, or you can actually take the whole plantlets also. Okay, now in short, organogenesis, it is nothing but the formation of new organs. Now what kind of organs we're talking about over here? It can be shoot, root, okay, and different parts of the plant. Now, in plants, development is the process that results in a functional mature organism which will include all the events during the life of the plant that produce the body of the organism. So in short, they're just talking about the development of the plant having all the shoots and roots, okay? And it provides the capacity to obtain food, to reproduce and to deal with hazards of its environment. So everything is included in the development process. So therefore, organogenesis is a process that is in some way unique to plants. Okay, so the shoots, let's talk about the shoot over here. The shoot buds are the monopolar structure over here. Okay, and they give rise to leaf and primordial and the apical meristem. So if you see in the plant, you have got the stem, leaves are there. Okay, on the top, the meristematic tissues are there, primordial leaves are there. So from here, you get small leaves are there. Okay, and this portion is called the apical meristem. Now, the simulation of shoot bud differentiation in plants, it depends on many factors. Okay, so Skoog showed. So if you remember this person, he has also given the MS media, which is used for the plant tissue culture. So he showed that auxin could stimulate rooting and inhibit shoot formation. But if you add cytokinin, you can also initiate shoot formation. Please remember. So this is important. The other factors which will affect the organogenesis are the size and the source of the explant. Okay, so light intensity also plays an important role. Of course, plant needs light in order to carry out photosynthesis. So high light intensity will be inhibitory for the shoot bud formation, and that's why whenever there is a high intensity of the light, or even if the sunlights are present, the plants will not grow. It will not become more taller. It will start lateral branching. Okay, so even the quality of light will have the effect. For example, as blue light shows or it induces shoot formation, while the red light stimulates rooting in case of tobacco plants. All these studies were done in the tobacco. So guys, this is also important. If you can remember that blue light is helpful for the shoot, and whereas red light is helpful for the roots. Okay, and then of course the optimum temperature will vary depending on the plant species you're working on. And the media what they use, it will be solidified using the agar, and it favors bud formation, although there are some reports about the development of leafy bud on the cultures grown as suspension. So if you see that, if you have in the conical flowers, agar is there. If you inoculate your plantlet or a small piece of your plant tissue, it will favors the bud formation. Okay, as compared to the suspension. So there's this video which I would like to share. I have taken from the YouTube only, and the URL has been shared with you guys. Just watch this video. So you can see that it is actually a time-lapse video of a cauliflower showing the organogenesis. You can see that this was the MS media. Agar is there. Small piece of the tissue was used. So you can see how nicely it starts forming the shoots. The leaf starts coming over there, right? So guys, don't think that it will be seen as this as soon as this. Okay, it's not like that. That is time lapse video, so it must have taken around months in order to get this kind of video. So medium also they have shared what all can we use, and of course the photography and all those people what they have done. Okay. So there are two types of organogenesis, indirect and organogenesis. So what do you mean by indirect? It means that whatever the explant you use, it will become callus, and then meristemoid, then it goes to the primordium. Okay, so it is that first callus is produced from the explant, so that's why it's indirect, because when explant is becoming callus, it means it is getting de-differentiated, and after that whatever phytohormones you provide, it is going to become the redifferentiation. Okay, then it will undergo the redifferentiation. So organs can then be produced from the callus tissue or from the cell suspension produced from the callus. So depending on that, you can actually carry out. So what they have shown is a small part of the leaf has been taken, and then it is actually callus is formed, and from the callus you can get many leaves. Okay, so it means what kind of phytohormones they must have used? Cytokinin. Now coming about the direct organogenesis, in this the callus is not formed. Okay, you can see the difference. So here from the explant, it is direct to the meristemoid and then to the primordium. So in many plants, subculturing of callus results in undesired variation of growth. Okay, so we'll talk about somoclonal variations later in the upcoming videos. And in order to avoid this, like the undesired variations, so what happens, even though they are the clones, but again they will have the differences. So in order to avoid this, direct regeneration of the explants into plantains can be tried, and that is what is called direct organogenesis. Okay. Now, how this can be achieved? By altering the hormonal combination of the culture media, of course. And this is another video which has been, made by these people working over there in the Spain. So very nicely they have shown the electron micrograph images. Also they have shown when a callus is formed and when the callus gets converted into shoot and root. So all those things can be seen in this video. Okay, so let's talk about the indirect. You can see that over here, having the different combination of auxin and cytokinin, different callus were formed. So this is the first week, only leaves are there, which is present on the media. Then on the week third, you can see it starts growing. Okay. So regenerated shoots are there, callus is there, and first leaf, which you get from the callus. Okay. So from here also you can see this is the electron micrograph images of the callus. So you can see how it is the apical meristem will appear from the disorganized callus. So actually it's a callus. Okay. You can see the cells are not arranged properly. So moving further, so they have got the spongy callus tissue is there. This portion has been characterized. See here, small pieces are there as compared to this big one, right? So they are more compact. And here it is the cuticle layer of the plant which is present. Okay, so if you see over here the different areas of the callus, again, so here it is linked by the pectin projections. You can see the pectin projections are here. Now this is the epithelial tissues where they have shown the arrow. And these are the columnar apices. Now this is the root. Again, another image of the rootstock only. And this is, just remember, this is after the callus, and from the callus, the regeneration of the root. So those images are there. So these are from the leaves. Again, the shoots are forming. Now talking about the direct organogenesis. So these are the plants they have grown from the using the method of direct organogenesis. So if you see, if you're very well known with the electron micrograph studies, then you can understand what is happening in the images. Okay, see these are the glandular hairs. They are the apex emerging from here on the top. Yeah. Again, the image of the apex is shown over here. Then these are the leaf primordia. Then here the shoots are getting regenerated. Now this is for the roots. So this is the plant, the tree they have selected. You can see over here, they are actually growing the shoots and roots, and they are doing everything from the root. So actually the root portion was taken, and from the root, they have added the phytohormones in order to get the shoots. So this is the origin. And then at the apex, you can see very beautifully it is emerging. Now, from here, the shoots, you can see the primordia, the leaves are actually getting regenerated. So root organogenesis is also called rhizogenesis. This is in the, see, solid media they have shown. Now then the, so this and all, if you remember your cortical cells and those xylem and phloem cells and also, then you can study all these images. So thank you guys for watching this video. I hope this was helpful, and I'm sure you must have got the idea how exactly the plant tissue, in plant tissue culture, the organogenesis is being carried out. Okay, so stay tuned with us so that you can study more and prepare for your exam. Thank you guys.